Hello everyone, welcome to your prep screencast for chapter 18, which is on the heart. So we see a cadaveric heart here, and really we just see the inside of the ventricles. So we're going to talk all about the heart in the coming slides. Okay, so first thing we want to understand is the two types of circuits that we have within our body. So here pictured I have the heart, and our right side is really working toward pulmonary circuit, meaning we have deoxygenated blood coming into the heart here at the superior vena cava and inferior vena cava, filling up the right atrium, moving down our right atrioventricular valve. And don't worry, we'll go through these names again for the valves and the chambers. Then that takes us to our right ventricle, and then we're going to go through an area called the pulmonary semilunar valve to our pulmonary trunk. Now, what that does is it's going to split into pulmonary arteries. We only see it on one side here, but realistically, we're going to have one go to the right side and one go to the left side. Then that goes and branches into smaller arteries, eventually capillaries within the lungs, and we are picking up the oxygen that we have breathed in from our environment down into our lungs. So that blood then gets put into that oxygen gets put into our blood, and then we carry through our capillaries to our venules and then to our pulmonary veins. And again, we're going to have this both on the right and left side, and bring it back to the heart. This is now when we are going to start our systemic circuit. So we get oxygenated blood to the left atrium. We're going to go through our left atrioventricular valve into our left ventricle, and that is our last chamber of the heart. After that, we're going to go through an aortic semilunar valve and through our aorta, and basically our aorta is going to pump oxygenated blood to our entire body to give that oxygen to our tissues. Okay, so let's just quickly talk about each of the chambers of the heart. We have 4 chambers of the heart: the right atrium, right ventricle, and left ventricle. And between our right atrium and right ventricle, we're going to have something called a atrioventricular valve. Now I'll reference this in the coming slides as well, but I just wanted to show you that valve sitting in between these chambers. We already mentioned that our right atrium is going to receive deoxygenated blood. Now this is coming from our systemic circuit. And I know some of you may be thinking, well, wasn't the systemic circuit on the left side? Yes, that is correct. This left side pumps oxygenated blood to our entire body, all of our systems within our body. But then once that tissue gets that oxygen, we have to return the deoxygenated blood back to the right side of the heart. So that's why we say returning from the systemic circuit. So then from here, we're going to move into our right ventricle. And like we said, this will go through our pulmonary semilunar valve, through the pulmonary trunk to our lungs. And then we get our oxygenated blood coming to our left atrium. So this is receiving blood from our pulmonary circuit, goes through our left AV valve or atrioventricular valve to our left ventricle, and then that's going to pump blood to our systemic circuit through this aorta. Okay, before we get into the different parts of the heart, we want to talk about the layers of the heart wall. And so we really have 3 main layers, but our first layer, which I'm going to oh, I forgot I did this in a different order. Okay, let's go back. So our first layer, so most superficial, is going to be something called the fibrous pericardium. Remember, peri means surrounding, cardi meaning heart, and meaning pertaining to. So really this is a big sac that covers the heart, and you guys will see this really well in the anatomy lab. So I'm excited to take you there and show you this actual tissue. Now, our pericardium is made up of 2 different layers. We have the fibrous pericardium, which we see labeled right here, and also serous pericardium. Now, we learned this back in chapter 1, so let's review real quick. A serous pericardium, that means it's broken down into 2 different layers. We have a parietal layer, which if you remember, parietal means wall. So this is lining the wall of our pericardial sac, and then viscera means organ. So this is right on the organ itself, really. So here we see the epicardium, or also known as our visceral layer of our serous pericardium. And below that, we have our myocardium, which is the heart tissue itself. So that really makes sense why we have our visceral layer here. And of course, between our parietal layer and our visceral layer, we have a space that's built, and that's going to be our pericardial cavity. Now, we already said our myocardium is the heart muscle itself, so I have that outlined here in purple. And then on the inside, the innermost layer is going to be our endocardium. It's going to be continuous with our endothelial lining of our blood vessels, which we'll look at that several blood vessels on the heart as well. And it's going to line each of the chambers of the heart, so our two atria and our ventricles. So let's do a quick overview of some structures here of the heart. We've sliced the heart in half here so you can see just about everything, and this is a frontal section. So first of all, when you get into the lab, I always tell my students to sap the heart. Now, what does that mean? That means you look for S for the superior vena cava, A for the aorta, and P for the pulmonary trunk, okay? So if you sap the heart, you really help orientate yourself to where all your other structures are. So our superior vena cava, if we draw a line down the superior vena cava, we should be able to find our inferior vena cava as well. These two large veins are going to bring in deoxygenated blood to the right atrium. Our right atrium has a couple different structures on here. So we're going to have some pectinate muscle lining our right atrium, and I'm going to show you a cadaveric picture of what pectinate muscle actually looks like in a moment. And then on the inner wall here, on our interatrial septum, which inter meaning between, atria referring to the atrium, septum meaning wall, so this is between our two atria. Here's our left atria. We're going to have a depression here, the fossa ovalis. Remember, fossa means depression, ovalis is referring to an oval shape. That's going to be remnant of a hole that was in our heart when we were a fetus, so that blood could shift back and forth between the atria. So we'll look for that in lab as well. Okay, so our deoxygenated blood here is going to move through this valve, and I kept referencing this as an AV valve, or our right atrioventricular valve. It also has another name, and that is the tricuspid valve. So you can use either term, right atrioventricular valve or tricuspid valve, and that's because we have three cusps here that make up this valve. These cusps are attached to chordae tendineae. They're just these long cords that are tendinous that attach to a cone-shaped muscle down here called the papillary muscle. So all these structures are located within our ventricle, and this is the right ventricle here. The deoxygenated blood pools into this ventricle, then goes through our pulmonary valve or pulmonary semilunar valve, through a pulmonary trunk, and then we'll go out through the left and right pulmonary arteries. Now notice these arteries are in blue. Remember that an artery just means that it's sending blood away from the heart. Veins always bring blood back to the heart. And it's blue because our blood is going to be deoxygenated here still, because these pulmonary arteries are taking the blood to the lungs. Once we go to the lungs, this blood becomes oxygenated and it comes back through these two veins here called the right pulmonary veins, and then we have two veins on the other side, so these are the left pulmonary veins. They all filter into the left atrium and then go through our left atrioventricular valve. Now this valve has three different names, lucky for us. So you can reference this as the left atrioventricular valve, mitral valve, or bicuspid valve. Bicuspid because we only have two cusps here. These are also attached to chordae tendineae, which attach to the papillary muscles in the left ventricle. Now notice how this left ventricle is much, much thicker, about 2 to 3 times thicker than our right ventricle. And that's because we got to pump this blood to the entire body instead of just the lungs, the pulmonary circuit, like we do on the right side. So this blood pools in here, and then we're going to go through our aortic valve or aortic semilunar valve. That goes through our ascending aorta, aortic arch, and then down our descending aorta. This continues down through our thorax, abdomen, into the pelvis, well, really it's going to split into two arteries at the level of lumbar vertebrae number 4. So we'll talk more about that in our blood vessel lecture. So this is the heart put as a whole, so we see it covering. The only thing I want to mention here is that we have these ear-like appendages called auricles. These will allow for extra storage of blood, and they're going to kind of lie over each atria. So this is the right auricle laying on the right atrium. This is our left auricle of our left atrium. Also, at the area where we have our aortic semilunar valve, right after that, we're going to see an opening for our coronary arteries. So these are the coronary arteries. Our coronary arteries help supply oxygenated blood to the heart itself. Now, once that heart muscle uses up the oxygenated blood, it takes the deoxygenated blood and puts it into these cardiac veins, which filter all the way back to our posterior view here of the heart now, but they're all going to filter into our coronary sinus, and this coronary sinus goes and brings the blood into the right atrium so that we can pump it through the heart again. So more detail on that in our class lecture on Thursday. Okay, so here is a view of our right atrium. This is that pectinate muscle I was telling you about in the right atrium, and then we have that fossa ovalis as well, and this is pointing out for you the right auricle. And by the way, our auricles also have pectinate muscle within it. Now, I also want to make you aware that this pectinate muscle is also called musculi pectinati. Similar name, but just a little flip-flopped. And here's that cadaveric picture I was also referencing that I would show you. So here's this pectinate muscle right in here of our right atrium. Okay, what was I going to show you here? Okay, for some reason those arrows, oh, there they go. This is our ventricle here, I believe the right ventricle, since that ventricular wall is a little bit thinner. And I just wanted to show you the structures that you're going to be taking a look at in lab. Here they've cut open the right atrioventricular valve. We have one cusp here, and another one here. And all of these are attached to the chordae tendineae, which attach to the papillary muscle. So when they contract, they're going to pull on these chordae tendineae to help functioning with our cusps here of the valve. And then the rest of this webbing-like structure here is going to be called trabeculae carneae. We only find trabeculae carneae within the ventricles. And when I first took anatomy, one of the TAs in there said it reminds him of Taco Bell, so that kind of stuck with me. So I would always think carne, they're spiced beef. So I don't know if that helps or hurts, but there it is. Let's move on. This chart is really great because it kind of gives you an overview of where the blood is coming into the heart and leaving the heart from. So let's just follow this along. We have our superior vena cava, inferior vena cava, and they don't have it pictured here, well, just a little opening, but the coronary sinus will all bring deoxygenated blood into the right atrium. From the right atrium, we're going to move through that tricuspid valve or right AV valve into the right ventricle. From there, we go into our pulmonary semilunar valve, into our pulmonary trunk, and out to the right and left pulmonary arteries. That goes to the lungs. That blood becomes oxygenated. We come back through right pulmonary veins and left pulmonary veins. Whoop, I did that backwards. Right pulmonary veins, which would go all the way here, and left pulmonary veins to our left atrium. And then we'd go through our bicuspid valve or left atrioventricular valve, or sorry about that minor break right in there. So the blood would pool into the left ventricle. And then from there, we're going to go through the aortic semilunar valve, which you could barely see here. That aortic semilunar valve is going to lead us to the ascending aorta, aortic arch, and then the descending aorta. And through the aortic arch and the descending aorta, we are really sending oxygenated blood to the rest of our body through the systemic circuit. Now, we already learned about skeletal muscle. Let's learn about cardiac muscle. So cardiac muscle, if you remember from our chapter 9 lecture, is going to be striated. We have more short branched cells, so you kind of see a little bit of branching here in this bottom photo. And we're also going to see that they're very interconnected. We'll talk about the interconnectedness in a moment, but first we need to mention we have 1 to 2 nuclei, which is centralized within each muscle cell, and we have a ton of mitochondria within our cardiac muscle. That's because our cardiac muscle only relies on aerobic pathways for its contraction. So our mitochondria are going to take up about 25 to 35% of our cell, and the rest of the volume is composed of our familiar Z discs, which you can see here in like the light blue here, and our A bands spanning from here to here, and our I bands would be the lighter portions. We still have our T tubules. They're going to be a little bit wider than our skeletal muscle, but they are less numerous, and these are going to line up with our Z discs like you can see here. Our SR, sarcoplasmic reticulum, will be present still, but they're a little bit more simpler. We don't have those terminal cisternae that would line up with our T tubules, so we really don't have any triads. Now, the other thing that separates cardiac muscle from our other two types of muscles is the intercalated discs. And I'm just going to go back to show you the intercalated discs here on this histology slide. So we got our intercalated discs here, and what those are, they're junctions between our cardiac cells, and within them we have our gap junctions, which you see pictured here, as well as desmosomes. The desmosomes, are helping to hold our cells together so that they don't separate during muscle contraction. And our gap junctions are going to allow the ions to pass easily from cell to cell so that the cardiac muscle is all going to contract together. And we call this functional succinctium. And we have two different types of muscle cells within our cardiac muscle. We have contractile cells, which are probably as similar as can be to our skeletal muscle. So we see them pictured here with their sarcomeres, and these are representing our mitochondria. And this is representing our skeletal muscle, where we would have our myocytes as well. And then what's different about cardiac muscles, we have our pacemaker cells. These pacemaker cells are also called autorhythmic cells, and these are non-contractile cells. And what's special about them is that they spontaneously depolarize. So they don't really need nerve innervation. They're almost like self-stimulating, and they send a signal to our cardiac muscle to contract. And that is going to, I'm going to skip this table for now. And those cells are going to lie within areas of our SA node, which I'm going to fast forward to show you where that is. So these next structures help make up our conduction system of the heart. We have our SA node, sinoatrial node, our atrioventricular node or AV node, our AV bundle, and then that goes to our left and right bundle branches and creates our Purkinje fibers, or also known as our subendocardial conducting system. So we're finding those non-contractile cells within these SA nodes, AV nodes, and all that. Okay, so let's compare skeletal muscle and cardiac muscle. They're both striated. Our cell shape is going to be a little bit different, and skeletal muscle was nucleated. Cardiac muscle was not. Also, cardiac muscle will have gap junctions, will contract as a unit. Both skeletal and cardiac muscles will have T tubules and SR. They're going to be a little bit different. Our source of contraction is still calcium for both, although in skeletal muscle it's only from our SR, whereas in the cardiac muscle we're also pulling calcium from our extracellular fluid. We'll have troponin in both because we have sarcomeres in both. We have special pacemaker cells present in our cardiac muscle, which we just talked about. Tetanus is going to be possible in skeletal muscle, but not in cardiac muscle. If you remember, tetanus is going to be when the muscle fiber is stimulated so rapidly that it doesn't relax at all between stimuli. And so that's why we only have it in skeletal muscle. And then our supply of ATP is going to be aerobic and anaerobic in skeletal muscle, but only aerobic in cardiac muscle. So we're going to talk more about this in class. I'm just going to pass forward through. We're also going to learn how to understand what EKG readout is, or ECG. And so these are the different waves. I'm just going to talk about this in class, but wanted to show you that we'll be discussing this in class. and then we have our mechanical events of the heart. So you may have heard of systole and diastole through various medical talks, or if you visited the doctor and had to discuss your blood pressure. So whenever we have our systole, that's going to be our period of heart contraction. And then what happens is our AV nodes are going to close up. And then when we have diastole, those AV valves are going to open up and our semilunar valves are going to shut. Basically, these two put together is going to give us our blood flow through the heart during one complete heartbeat. And we have our two sounds of our heart, the lub-dub that we're used to hearing when we listen into a heart. The first sound is when our AV nodes are going to start ventricular systole. So that means our ventricles are contracting. And the second sound, the dub, is going to be the closing of our semilunar valves and the beginning of our ventricular diastole, which is going to be when our ventricles relax. And we have a pause, which is going to indicate heart relaxation between the lub and the dub. And these are all the different areas where we can listen in for heart noises. And the last thing we're going to talk about is cardiac output. This is going to be the volume of blood pumped by each ventricle in 1 minute. And we can calculate our cardiac output by taking our heart rate and multiplying it by stroke volume. Now, heart rate is going to be the number of beats per minute, whereas our stroke volume is going to be the volume of blood pumped out by one ventricle with each beat. And this correlates with the force of contraction. So our cardiac output is going to be our heart rate, which typically is going to be about 75 beats per minute, and stroke volume, which a normal one is going to be 70 milliliters per beat, and that gives us a normal cardiac output of 5.25 liters per minute.